Hello. Thank you for joining me today in our continuing flow cytometry webinar series. Today's webinar will cover DNA content cell cycle analysis using flow cytometry. Previous webinars are available on our website for viewing, and this webinar will also be available in about 2 days. My name is Jolene Bradford. I work in the research and development of flow cytometry systems located in Eugene, Oregon. This is the home of molecular probes. Before I start the presentation, I'd like to make a few announcements. First, you can submit questions anytime during the presentation. Just use the Ask a Question tab on the left side of your screen. I'll try to answer as many questions as time allows at the end of the presentation. It's likely that I'll answer some of the questions after the webinar has ended. So now let's begin. Today, we will define DNA content cell cycle, look at reagent options available for cell cycle analysis in living cells, and additional options available for fixed cells. I'll spend the majority of time reviewing technical considerations for successful testing and offer numerous tips and tricks. The eukaryotic cell cycle, or cell division cycle, comprises a series of events that are involved in the growth, replication, and division of cells. Cell cycle describes the progression of a cell through a cycle of division. There are cytoplasmic events, and there are nuclear events. G1 cells, or cells in the growth phase 1, induce growth by first making RNA and proteins, but the DNA content does not change. In the S phase, or synthesis phase, DNA synthesis proceeds with DNA replication, and cells have variable amounts of DNA. Cells in growth phase 2, or G2, have completed DNA synthesis and continue to grow and prepare for mitosis, with DNA maintained at double the original amount. In the M phase, or mitosis, nuclear division and cytoplasmic division give rise to 2 daughter cells. After completion of the process, each daughter contains the same genetic material as the original parent in approximately half of the G2 level of cytoplasm. Cells can also exit from the cell cycle and enter a non-dividing stage, G0, where they can enter the cell cycle with proper activation. Often in talking about cell cycle in flow cytometry, we describe the distribution of a population of cells in the different nuclear phases. DNA content analysis is one of the most widely used applications in flow cytometry. During the S phase, cells undergo DNA replication, so that they enter the G2 phase, they have twice the normal amount of DNA. Looking at the amount of DNA within the cell indicates the proliferation state of the population and can be used to examine the effect of drugs that affect the cell cycle, something that is particularly important to development and cancer research. In this approach, cells are incubated with dyes that bind stoichiometrically to DNA. This means that the dye binds in proportion to the amount of DNA present in each cell. DNA content distribution in flow cytometry gives a single time point measurement. Using linear fluorescence, if the staining is stoichiometric, the fluorescence of the 4N cells will be exactly twice that of the 2N cells. Under ideal staining conditions, all cells with the same DNA content are expected to be uniform in staining. However, in practice, cell populations are represented on frequency histograms with peaks of various widths. Variation results from differences in staining, dye loading, instrumentation, and none are perfect. So this shows the histogram from the instrument on the left. And you can see it's difficult to accurately put a marker on the different phases because there are 3 overlapping distribution curves. DNA content histograms require mathematical analysis in order to extract the underlying G1, S, and G2 phase distributions. Method to derive cell parameters from DNA histograms range from simple graphical approaches to more complex deconvolution methods using curve-fitting algorithms with debris and aggregate modeling. There are many dyes that can be used for labeling of nucleic acids, which cross a wide range of excitation and emission wavelengths, as listed here. Many of these can be used for measuring the DNA content of cells, either in living cells or in cells which have been fixed. With so many choices, it becomes difficult to choose which dye to use. You can look at the excitation emission of the dye and know how compatible it is with the instrument you have to measure it with. Using a fluorescent spectroviewer is helpful in looking at overlapping emissions. You'll also need to understand what application you want to use and if the application requires DNA specificity, or if you need to use this with live cells or fixed cells. Cells which have the ability to penetrate an intact cell membrane to stain nucleic acids are called cell-permeant or live cell dyes. These can be used to determine the DNA content of living cells. The dyes listed here are also DNA selective, meaning they do not label RNA. All of these cell-permeant dyes are essentially non-fluorescent, cell-bound to DNA. With cell-permeant DNA selective dyes, live cells are incubated in the presence of the dye, and no additional treatment is necessary. Although most useful with living cells, these dyes may also be used with fixed cells. Hurst has been around for decades and has been for a long time the only choice for live cell cycle studies. It's a great dye, but it does require UV excitation. And now we have other choices with the dye cycle stains that allow live DNA content measurements using visible light. A live cell staining does allow for the possibility of sorting based on DNA content. In this example, 3T3 mouse fibroblasts are stained with 10 micromolar dicycle orange and sorted into the G0, G1 and the G2M phases of the cell cycle. You can see the post-sort verification of the sorted populations, and subsequent growth of these sorted populations is verified by showing the growth pattern after 3 days of growth. Here is another example, this time using vibrant dicycle ruby. Jurkat cells were stained with either Herx, DRAC5, dicycle ruby, or no stain as a control. The cells were sorted on a fax aria and then cultured, and Brightfield images were taken after 8 days in culture. Clearly, DRAC5 is cytotoxic to cells, there is no growth, while dicycle ruby behaves more like the Herx, showing the characteristic grape clusters of growing Jurkat cells. So it's reasonable to assume that any treatment may cause some amount of cell death. Here, dicycle ruby produced very similar results to the gold standard Herx. Here is an example of one of the uses that can be done with a live cell cycle dye. So this is taking human whole blood that has been treated with ammonium chloride to lyse the red blood cells, and then labeled with a CD45 APC direct antibody conjugate and with vibrant dicycle green. The white cells show the 3 populations of neutrophils, monocytes, and lymphocytes by the CD45 versus side scatter plot. You can simply gate on the population of interest and then look at the DNA content. While these cycle plots are not very interesting because the normal cells of the blood are not actively cycling, you can imagine doing this with bone marrow or peripheral blood where there is an abnormal cell population. You can also use the internal normal diploid cells as an internal standard. Protocols for labeling with these live cell dyes are very simple. They are DNA selective, so there is no need to add RNAs. The labeling is very simple. You have cells in suspension. You add the dye, you incubate, and then you acquire. And it's very important to acquire without washing. Impermeant nucleic acid dyes penetrate cells with a compromised membrane to stain nucleic acids, but do not cross the membranes of live cells. So these can be used to identify dead cells in a population, or they can be used to quantitate DNA content in fixed cells. Traditional stains like Propidium iodide and 7-AAD are now supplemented with newer options from the Cytox series that take the measurement off these traditional detectors and make wide use of a wide range of excitation sources. So viability is often referred to as the integrity of the plasma membrane. Cells that successfully exclude an impermeant reagent, such as Propidium iodide, are considered viable. Many of these same stains can be used for DNA content measurement in fixed cells. So an impermeant nucleic acid dye can have two uses. First, as a way to identify dead cells in a mixture of live and dead cells, as in the plot on the left. Your cells were labeled for only 5 minutes with Cytox Advanced stain and run with a 488 excitation source. You can see there is a clear distinction of live cells and dead cells. Second, DNA content analysis can be determined on fixed cells with the addition of RNase, and this is seen in the plot on the right. For DNA content measurements, be sure to use linear scaling, while with dead cell identification, log scaling is used. The fixation has two functions. First, it preserves the cells by preventing lysis and autolytic degradation. And second, it makes the cells permeable, and thus their DNA is accessible to the fluorochrome-binding DNA dyes. Precipitating fixatives, such as methanol, ethanol, acetone, are preferred for single-color cell cycle staining. Disadvantages include that epitopes can be destroyed, and this does induce more cell aggregation. Cross-linking fixatives, such as aldehydes, can have a harmful effect on the stoichiometry of the DNA staining, but fewer epitopes are destroyed and less cell aggregation is induced. You'll need to choose the best fixation method for your particular application. Our FX cycle stains are useful for DNA content measurement in fixed cells. FX cycle violet uses the 405 laser, while FX cycle far red uses the 633 red laser. FX cycle PI RNase can be used with a variety of lasers, the 488, 532, and it is provided in a pre-formulated solution. These do allow the use of the common blue laser or other markers. In this example, both FX cycle far red and FX cycle violet are used to label DNA content in a multicolor application with phosphohistone H3 detected with an Alexa Fluor 488. This identifies the cells in the mitosis phase. By using lasers other than the common blue laser, the use of FITC and PE markers can be combined with DNA content. While DNA content may seem like an easy test, it is often one of the most challenging applications to master. Many things can influence the measurement. Instrument performance, sample preparation, sample acquisition can have a significant impact on the cell cycle measurements, and the manner of data analysis can also affect the measurement. I'm going to review each of these in more detail. For any flow cytometry experiment, it's critical to understand the excitation and emission profile of the fluorescent reagents you are considering using, and then pairing that with the information with the configuration of the instrument you have. Our fluorescent spectroviewer can help you with the selection of dyes that fit your instrument, and here I'm showing the excitation and emission profiles of FX cycle far red and FX cycle violet dyes. The FX cycle far red uses the APC detection channel of the red laser, while the FX cycle violet uses the Pacific blue detection channel of the violet laser. Properly aligned optics of an instrument are especially important when doing DNA content cell cycle testing. Remember, we're not just looking at positive and a negative response, but we're trying to distinguish peaks of differing DNA content. And the lower the CV, the better. A properly aligned laser hits the cells in the center of the stream, while a misaligned laser will not be centered on the cell stream, and thus a greater variation will result. And this is seen with increased coefficient of variation. In this example of alignment beads run with a properly aligned laser, as on the left, and a misaligned laser on the right, you can see that on the right there is a high degree of imprecision, or an increased percent CV. It's recommended that you monitor your instrument daily. Having a single cell suspension is also very important in staining. Cell clumping or aggregation limits the accessibility of the dye to the cells. Cells and clumps will not stain consistently the same as single cells. And the dye will penetrate the outer cells in the clump, but will penetrate cells and the inner cells in a varying amount. Removing aggregates and clumps before analysis is highly desirable and is preferred when possible over any other method of gating or analysis. So before staining, there are some things that you can do. You can filter your cells through a 35 micron mesh filter to remove the larger clumps. You can do a process called trituration. So this is taking your cells and aspirating them back and forth through a very small needle, such as a 25 or 26 gauge. It may not be the best approach for delicate cells, however. You can also remove or decrease the calcium concentration in the suspension buffer. You can also try adding a bit of trypsin. Remember that sample processing for a particular cell or tissue should be optimized, and different recoveries and CVs may be obtained dependent on how sample processing is done. Laser stability is necessary. Please follow the instrument manufacturer's recommendation for proper laser warm-up time. One good way to monitor stability of the system is to use a fluorescence versus time plot. The top plot is a fluorescence versus time, and it shows an example of a steady laser, and this is what is expected. The lower left blue plot shows the same parameter, but where the CV is very large. This probably represents an alignment issue. And the red and violet plots are demonstrating laser instability, and there's either a problem with the system or the laser has not warmed up fully. Fluidic stability is also important. And again, you can monitor the stability of the system using a fluorescence versus time plot. Top plots demonstrate cell cycle staining where a steady fluorescence that does not change over time is seen. There's no drift upward or downward, and there's no breaks. The bottom plots demonstrate fluidic problems. A plug would cause a period of no collection, and variation in fluidics, as seen in the right lower plot, may result from changes in a flow rate, which could be due to a plug or a partial plug. Unlike high-affinity antigen-antibody binding, DNA binds with low affinity, and because of this, it exists with equilibrium in the solution. And it is sensitive to dye concentration and cell concentration. So the quality of the histogram depends on cell concentration. In this particular example, I have increasing cell concentration kept with a constant dye concentration. In this example, the voltage for each plot was not kept constant, but I could put the plots on scale. You can definitely see that the quality of the data changes with cell concentration. So especially within the same experiment, it is important to have the same cell concentration with each determination. It's also sensitive to dye concentration. So here we see an example of increasing dye concentration with constant cell concentration. In this example, the instrument settings were kept constant for all of the samples. And you can see the quality of the histogram depends on the dye concentration. The percent CV will increase at non-optimal dye concentration, and the position of the G0, G1 peak will shift. So again, it is especially important within the same experiment to have the same dye concentration in each determination. Lack of instrument linearity can produce non-standard G2, G1 ratios and can have a significant impact on DNA index values. It can also affect the aggregate estimates with the modeling software. Instrument linearity should be checked on a regular basis. One way to do this is using beads with varying fluorescence. There are many products available for this, and some instrument performance tracking measures the linearity for each fluorescence detector. Especially for the live cell dyes, temperature may affect the results. And again, you need to optimize this for each dye and labeling condition. Here, the top plots show Herxt at 37 degrees and room temperature. And here you can see that 37 degrees is really required and room temperature is not recommended for a DNA content histogram. The bottom plots showed dicycle green in HBSS at different temperatures. So again, it's important to use temperature as one of your optimization tools. Incubation time is also important. A longer incubation times sometimes may improve staining or give the ability to use a lower concentration of dye with a longer incubation time. So on the top, we have dicycle violet. At 30 minutes, the histogram has improved. On the lower, we have dicycle orange at room temperature. And again, the longer incubation time of 60 minutes is preferred. So remember to optimize your labeling conditions. If an insufficient number of cells are acquired for DNA content histogram, the accuracy of the analysis is impaired. Above 10,000 events per histogram, S-phase measurements are highly reproducible. Below 10,000, and especially with fewer than 1,000 events, the accuracy of the S-phase measurement deteriorates substantially. A minimum of 10,000 events in the DNA content cell cycle analysis has been recommended by the DNA Cytometry Consensus Conference Guidelines, although detection of DNA aneuploid populations may be possible from histograms with fewer events. Accurate S-phase analysis may require more than this if the proportion of aneuploid cells is low or if the debris is high, and the total number of cells should increase. Verity Software House has found that simulation studies indicate for an accurate S-phase estimates, there should be an average of at least 100 events per channel between the lowest G1 and the highest G2 of the histogram. When performing live cell cycle analysis, it's a good idea to remove the dead cells from the analysis. Remember that these live cell stains will enter both live cells and dead cells and will label them both. Here is an example showing a population containing a significant number of dead cells. And I've used Cytox Blue to identify the dead cells, remove those, and gate only on the live cells for the DNA content measurement. This improves quality of the data. The DNA content is no different in the dead cells, but the difference is due to the conformational changes in DNA as the cells are dying, and this affects how the DNA is stained. The elimination of dead cells when doing a live cell assay may be some new concept because using the classic dyes that require cells be fixed, there can be no distinction between the live and dead cells. With testing of live cells, one needs to consider this. There's a number of ways to gate out dead cells, and I've shown here using impermeant nucleic acid stains. There are other options like using an amine-reactive dye. So demonstrated here is a forward scatter versus Cytox blue. Sorry, dicyclone green versus Cytox blue, or forward scatter versus Cytox red, and then pulling out just the live cells. I mentioned the amine-reactive dyes. These are the live dead fixable dead cell stains. These are based on the reaction of a fluorescent reactive dye with cellular amines. The reactive dye can permeate the membrane of a compromised cell and react with free amines both on the interior and on the cell surface, which results in an intense fluorescent staining. In contrast, only the cell surface amines of viable cells are able to react with the dye, and this results in a dim staining. This discrimination is completely preserved following aldehyde fixation. With conventional flow cytometry instruments using hydrodynamic focusing, increasing the flow rate results in greater variability of results or increased CVs. In this example, labeled cells were collected at a low flow rate of around 12 microliters per minute, and this is displayed on the left. And then without taking the sample off the SIP and changing to a high flow rate of about 60 microliters per minute, the sample was acquired again, and the results are seen on the right. There is a clear increase of CV and decrease in overall quality of the histogram at the high flow rate. With hydrodynamic focusing, the sample is injected into a stream of sheath fluid where it is completely surrounded by the sheath. The sample core is then centered in the sheath fluid where the laser beam will interact with the cells. Based on principles related to laminar flow, the sample core remains separate but coaxial within the sheath fluid. The flow of sheath fluid accelerates the cells and restricts them to the center of the sample core. This process is known as hydrodynamic focusing. When the sample core is narrow, but with a low flow rate, the cells interact with the laser beam optimally and give a narrow distribution and low CVs. To increase the flow rate in hydrodynamic systems, the speed of the sample core does not change, but rather the width of the core within the sample sheath sample increases, which allows more cells to enter the core stream at any given moment. With a wider sample core, some cells will pass through the laser beam off-center and intercept the laser at a less optimal spot, creating variability. This results in a wider distribution of data and higher CVs are obtained. This limitation, the trade-off of speed and sensitivity, has largely been accepted in flow cytometry, and the need to use a low flow rate for samples such as DNA content, where very precise results are required, is well known. So here I'm showing an example of alcohol-fixed Jurkat T cells labeled with Propidium iodide staining solution containing RNase. On the left, you can visually see that an increase in sample rate leads to an associated decrease in data quality as measured by the percent CV. In addition, you can also see there is a significant change in the percentage of cells measured to be in the S phase of the cell cycle. For this reason, instruments using traditional hydrodynamic focusing produce their best cell cycle data only at the lowest flow rate. Acoustic focusing focuses the cells in the middle of the sample stream, no matter how wide the core is. With acoustic focusing, it doesn't matter how wide the sample core is. The cells are always placed at the very center using acoustic forces, where the cells will always interact with the laser in an optimal manner. This brings precision at all speeds and allows for collection rates at much faster rates than hydrodynamic systems. With acoustic cytometry, there is no trade-off of speed and sensitivity. Because of this, sample rates can also be increased dramatically up to 1,000 microliters per minute and give highly precise measurements. The same Propidium iodide-labeled Jurkat cells, as shown before, were also tested on the Attune acoustic cytometer at collection rates ranging from 25 to 1,000 microliters per minute. The plots on the left for these collection rates show there is little change to the data quality as measured by CV. In addition, there is little change in the percentage of cells measured B in the S phase of the cell cycle. So the Attune produces precise cell cycle data across collection rates, not just the low collection rates. So now any collection rate can be used for DNA content measurement when using acoustic focusing. Here is a side-by-side comparison of a sample acquired on a high-end conventional flow cytometer using a violet laser and a low sample rate setting. Approximately 15,000 total events were acquired on both conventional system and the acoustic system. Alcohol-fixed HL60 cells were labeled with the FX cycle violet, and you can see that the underlying cell phase distributions and the percent coefficient of variation is defined. The measurement using the acoustic system is highly precise. So how do aggregates affect cell cycle? So for analysis, we don't want two G1 cells analyzed as one G2M cells. And methods for eliminating or correcting for the presence of doublets or aggregates include gating and software modeling. With gating, you can gate out doublets and aggregates using a technique called doublet discrimination. And this has been the classic approach to distinguish aggregates by virtue of their altered electronic pulse shape in a cytometer using a height or area versus width or a peak versus area. Aggregates can be separated out. However, this approach has some limitations. It works best with hematopoietic cells or cells that are a perfect spheroid. It might be a significant percentage of the cells acquired, and you run the risk gating out cells of interest. Software modeling is an alternative approach for the detection and subtraction of aggregates by software algorithms. This debris and aggregate modeling depends on the analysis of either smaller sized debris or larger sized aggregates to determine the effect of the debris and aggregates on the correct S phase and G2M phases. In the ideal flow cytometry analysis, suspension of cells is free of aggregates. However, that's rarely the case. Here, this shows pulse processing approach, where the management of aggregation is determined by the altered electronic pulse shape. Again, this has some limitations. The signal processing gating to eliminate aggregates is only recommended if the aggregates are clearly and completely separated from the singlet population. This is usually true for tissue cell culture lines and for hematopoietic cells. This method eliminates the doublets and the aggregates from the data analysis, and it depends on the instrumentation, particle shape, and the operator's selection of a gate. Identification and correction for debris and aggregates can be done through complex software algorithms using probability modeling. This does not eliminate from the cells from the analysis, but identifies and corrects for them. So this is an alternate approach. The debris and aggregate modeling depend on the analysis of smaller debris sized, and it corrects for the presence of aggregates and debris. Neither approach is perfect, and what is currently controversial is whether both of these may be used sequentially. Ploidy is related to the number of chromosomes in a cell. Diploid, it refers to the normal 2N number of chromosomes, while aneuploid is an abnormal number of chromosomes, something that is not 2N. The medical community has not really achieved a consensus currently about the significance of ploidy and S phase and how to use it. Some of this is due to inconsistent guidelines and procedures between labs and between reporting systems. Tumors often have an abnormal DNA content. In this case, ploidy may have a prognostic value. Tumors often also have an increased proliferation. And here the percent S phase may have a prognostic value as well. In the plot here, I'm showing a sample that has a diploid and a near diploid population. This is an example of a DNA-Tetraploid population. In this case, Jurkat T cells were treated with cytochalasin B. This is a cell-permeable toxin that inhibits cell division while allowing nuclear division to proceed. And this results in DNA polyploidy. Control cells on the left and the treated cells are on the right show that a tetraploid population is identified. This is dicycle violet, demonstrated with ModFit analysis. So returning to our question about which dye to use, remember your instrument configuration and select a dye that is compatible. Determine what application you need. Know if you need live cells or fixed cells. And if you're using fixed cells, understand the effects of your fixation method. Understand the specificity of the dye you have chosen, and above all, optimize the conditions for labeling, acquisition, and analysis. And that does bring us to the end of this presentation. I'd just like to remind you that Life Technologies offers many resources. The latest one is an updated flow cytometry mobile app. You can also find more technical resources by visiting our flow cytometry resource center. And I'd like to invite you to join our Facebook pages to share your questions and learnings with the communities. And I'd like to thank you for your attention. And I'd like to remind you that this webinar, as well as previous webinars for both imaging and flow cytometry, can be viewed by visiting our website. So I have one question here. Why can't you wash after adding a DNA stain? It's best to add a DNA binding stain as the last step in your protocol and not wash it out. The dye is essentially non-fluorescent until bound to DNA, so it shouldn't add any background. The dyes DNA binding dyes exist in an equilibrium with the dye in solution. The binding is non-covalent. And if this equilibrium is disrupted, then it can change the labeling. So this actually applies to both live and dead DNA binding dyes. So I have a question about what should I do first for optimization. I can say that with initial experiments, you can try a range of dye concentrations to determine one that gives you your best or optimal staining. This would be for a given cell type, a buffer, whatever your experimental conditions are. And it's really important to remember that within the same experiment that you have the same number of cells with each sample in your test and the same concentration of dye. I have another question here about combining a cell cycle with other assays and how to do that. And I can say that if you want to add a cell cycle stain with other stains for multicolor applications, that you'll want to first apply the other stains to the sample following the instructions for that particular labeling, which would include wash steps. But as the final step, add in the DNA binding dye and don't wash it out. It's also important in looking at multicolor applications to note that DNA binding dyes have a very broad emission. This is just in general. And that to label in a stoichiometric manner these dyes are very bright. And this means that they may use up more than one detection channel, and so you can see bleed through into unexpected channels. That's just something to be aware of with DNA binding dyes in general. You can also use a spectroviewer to help pull apart what you're doing and select the correct channels and combine your multicolor applications. There was another question about using about the application of side population. And so human and rodent stem cells efflux both the Herx 33342 and dicycle violet stains. And that's the basis for the side population technique. So you have to be aware if you have stem cells that you're labeling with dicycle violet or with Herx, that the dye can be pumped out. This efflux can be blocked with verapamil or some other blocking agents to prevent the dye efflux, and then you'll get an accurate DNA content analysis in these stem cells. The side population technique, for those of you who don't know, identifies stem cells and early progenitor cells in hematopoietic tissues, and the dicycle violet or Herx is loaded into your population of interest, and these cells subsequently pump out the dye with a membrane pump-dependent mechanism, and this results in a tail called the side population. All right, well, we will conclude this webinar. I would like to thank you for your attention, and if you have further questions, you can contact our technical assist line. Thank you very much.